Introduction. Welcome to today's lecture on the electron transport chain, ETC, a crucial process in bioenergetics and oxidative phosphorylation. In this lecture, we will explore the overview, structure, and intricate mechanisms of the etc. We will also delve into the role and functions of each complex involved, such as complex I, complex II, complex III, and complex IV. Additionally, we will discuss the importance of coenzymes, proton pumping mechanisms, ATP synthesis, and the regulation of the etc. and oxidative phosphorylation. By the end of this lecture, you will have a thorough understanding of the biochemistry and complexities involved in this fundamental process. The electron transport chain, located within the inner mitochondrial membrane, plays a pivotal role in oxidative phosphorylation. It consists of a series of protein complexes and electron carriers that collaborate to transfer electrons down a redox gradient, yielding energy in the form of ATP. The etc. comprises four main protein complexes: complex I, NAD dehydrogenase; complex II, succinate dehydrogenase; complex III, cytochrome BC1 complex; and complex IV, cytochrome C oxidase. These complexes are embedded in the inner mitochondrial membrane, forming a structural framework for electron transport. Bioenergetics refers to the study of energy flow and metabolism within living organisms. Oxidative phosphorylation, the ultimate goal of the etc., involves the coupling of electron transport with ATP synthesis. This process is driven by redox reactions, where electrons are transferred between electron carriers, causing a change in their oxidation states. During this process, energy is released and utilized to establish a proton gradient across the inner mitochondrial membrane. Complex I, also known as NAD dehydrogenase, is the initial entry point of electrons into the etc. It accepts electrons from NAD, which is generated during glycolysis, the citric acid cycle, and fatty acid oxidation. Complex I transfers the electrons to coenzyme Q, ubiquinone, while simultaneously pumping protons out of the mitochondrial matrix. This proton pumping activity contributes to the establishment of the proton gradient, known as the proton motive force, PMF. Complex II, or succinate dehydrogenase, complements complex I by directly accepting electrons from succinate, a product of the citric acid cycle. Unlike complex I, complex II does not contribute to proton pumping. However, its unique feature lies in its role as both a component of the etc. and an enzyme involved in the citric acid cycle. Complex II provides a crucial connection between these two metabolic pathways. Cytochrome C, a small protein located in the inner membrane space of the mitochondria, plays a pivotal role in electron transfer within the etc. It shuttles electrons between complex III, cytochrome BC1 complex, and complex IV, cytochrome C oxidase. This mobile carrier facilitates the intricate movement of electrons, aiding in the efficient generation of ATP. Complex III, or the cytochrome BC1 complex, mediates electron transfer from coenzyme Q to cytochrome C. While transferring electrons, complex III pumps protons across the inner mitochondrial membrane, further reinforcing the proton gradient. This complex acts as a crucial junction in the etc., facilitating the release of energy and ensuring electron flow towards complex IV. Complex IV, or cytochrome C oxidase, functions as the terminal complex in the etc. It receives electrons from cytochrome C and catalyzes the reduction of molecular oxygen to water. During this process, complex IV harnesses the released energy to pump additional protons across the membrane, further amplifying the proton motive force. The proton motive force, PMF, is a crucial component of oxidative phosphorylation. It encompasses the electrochemical gradient of protons generated by the proton pumping activities of complexes I, III, and IV. The PMF comprises two components: the electrical potential difference, delta psi, across the membrane, and the pH gradient, delta pH, across the inner membrane space. Together, these components drive ATP synthesis via ATP synthase. ATP synthase, also known as complex V, is an enzyme complex responsible for converting the energy stored in the proton motive force into ATP. It spans the inner mitochondrial membrane and consists of two primary components: the F0 subunit embedded in the membrane and the F1 subunit facing the mitochondrial matrix. As protons flow back into the matrix through the F0 subunit, the F1 subunit catalyzes the synthesis of ATP from ADP and inorganic phosphate. Coenzyme Q, commonly referred to as ubiquinone, serves as a mobile electron carrier within the etc. It shuttles electrons between complexes 1 and 3, facilitating the transfer of electrons from NAD and succinate to cytochrome C. This lipid-soluble molecule plays a crucial role in the electron flow and overall efficiency of oxidative phosphorylation. Thermodynamics plays a significant role in the efficiency of oxidative phosphorylation and the etc. The concept of Gibbs free energy dictates the direction and feasibility of electron flow. By understanding the redox potentials of electron carriers and their respective complexes, we can elucidate the favorable direction of electron transfer, ensuring the efficient generation of ATP. The etc. and oxidative phosphorylation are tightly regulated processes to meet cellular energy demands. Various factors, such as substrate availability, ADP/ATP ratio, and hormonal signals, control the rate of electron flow and ATP synthesis. Feedback mechanisms and regulatory proteins fine-tune the activity of the etc., allowing for precise control and adaptation. The electron transport chain and oxidative phosphorylation are intricate processes essential for energy generation in living organisms. The interconnected protein complexes and electron carriers work harmoniously to transfer electrons, pump protons, and create the proton motive force. ATP synthase then utilizes this force to produce ATP, the universal energy currency. Understanding the detailed mechanisms and regulation of the etc. provides insights into cellular metabolism and provides a foundation for studying various pathological conditions related to energy production.